हाँ जी, स्टार्ट कर दें फिर। मुझे जीआर ने बताया कि आवाज आ रही है, फुल शो
जी
हो
सर।
रही है।
जी सर, कर दें शुरू।
सर आवाज तो आ रही है बट बहुत स्लो आ रही है।
ओके ओके ओके, बिस्मिल्लाहिर्रहमानिर्रहीम। अगर एक स्टूडेंट माइक चला लेता तो मुझे साथ साथ पता चलता रहता, लेकिन नो प्रॉब्लम। तो हम कंटिन्यू करेंगे इंशाल्लाह। कंटिन्यू हम करते हैं। So we have only the chapter 8th in the midterm examination. So half of the chapter have already been taught to the evening class, and the morning class have been taught with the full chapter. And the title of this chapter is the genes, genomes, and the chromosomes. And this chapter will be start from the molecular cell biology book textbook. This is and written by Lodish, and this book is named as the Lodish Molecular Cell Biology. So these are some of the basic information I have taken from the previous book. So why we need to study about the nucleic acid and the genes and the there are some good reasons that the nucleic acid are the part of energy currency. They are structural part of some of the enzymes and cofactors and the metabolic intermediates, who the nucleic acid and the nucleotides also constitute the nucleic acid. Okay, so nucleic acid is the polymer of nucleotide, just like the amino acid make the protein. So nucleotide make the nucleic acid. So these are some of the examples. They are the part of energy currency. These are shown here. So they are the part of the coenzyme you can see here. And how the nucleotide, this is one of the nucleotide actually. Nucleotide is containing three things: the nitrogenous base, the ribose sugar, it is shown here in the green, and the phosphate part. Three things a nucleotide must contain, then it is called the nucleotide. If the phosphate group is not present, then it is called the nucleoside. Only ribose and the nitrogenous base is called the nucleoside. This is the nucleotide full molecule. So how one nucleotide is joined to the other nucleotide? So the OH through is bonded to the phosphate group of the next nucleotide and is making the phosphodiester bond. This is the phosphodiester bond that is joining two of the nucleotides. So these are some of the basic information. Once again, gene. What is gene? Gene is a segment of the DNA, the segment of the chromosome that is making the functional product. So the functional product of a gene can be a protein or it can be an RNA. Okay, I continue then. And the RNA. Okay, so all the information about the cell fate are stored into the DNA. These information are stored and are transmitted through the genes. And there are different types of RNA: the ribosomal RNA, the messenger mRNA, the transfer RNA. There are different types of RNA. So they are performing different functions. We have already studied in the previous classes. Okay, so some other basics: DNA is a double helix. Helix, this is called the helix. I try to make the helix for you. This is called the helix. But the DNA is double helix. DNA is double helix this way. DNA is a double helix. So DNA is double helix. DNA specimen. DNA, if we extract from the liver, lung, skin, hair, from the gums, from anywhere, if we extract the DNA, they must have the base composition. So in one individual, all the time, the DNA base pair, the base sequence is always same, must always be the same. But there is only one variation. There is one exception actually. The B cells, the plasma cells do have PLAS. Plasma cell do have the different composition of their DNA depending upon the antibody they are generating. So only the plasma cells do have the different DNA. Otherwise, all cells in one animal must have the same base composition. And the base composition of the DNA does not change during the organism's age. nutritional status are with the environment. So DNA bases are not changed. But under the cancer, some of the time the radiation can cause the mutation, but naturally they do not, they are not affected by the nutrition or by the age. So we are moving to the next slide. DNA of high
Sir, screen
eukaryotes
share नहीं
consists
हो
of
रही.
both unique and repeated sequences. Only 1.5% of the DNA is the coding. Is coding mean they are making the functional product? But in the book, Lodish, 2% of the DNA has been reported as a functional DNA. So you can read these information, basic information. So we are now on to the first chapter, first page, and the first picture of the book, Molecular Cell Biology. So you can see here the nucleus. You can see here the nucleus, and in between these partitioning, there is some vacant spaces that are called the nuclear pores. So these are called the nuclear pores. So the things are exchanged many times, especially the mRNA are exchanged with the cytoplasm, uh, and the nucleus through these nuclear pores. So these are loosely bound chromosomes. These loosely bound chromosomes, when they are unpacked, we come across this kind of you can see loops. These loops consist of 30 nanometer fibers. Okay, this is one. This is the you can see the highest level of packing is the chromosome. Then you can see the higher order chromatin folding comes. Then the loops appear. Loops. This is the lower arrangement, lower level of folding. First one, chromosome. Highest is chromosome. Then the chromatin folding. Then the loop is there, and then you can see here the chromatins. You can see here the nucleosomes. Nucleosomes are folded somewhere here, and several nucleosomes are here, and this is consist of 30 nanometer fiber. These are also the 30 nanometer fiber. These are the 30 nanometer fibers, and the 30 nanometer fibers, when they are explored more, so their packing appear in the form of nucleosome. Nucleosome consists of 8 histone proteins. How many proteins? 8 histone proteins, and 2 rounds of the DNA around the 8 histone proteins is making the one nucleosome. Is making the one nucleosome. 8 proteins and 2 rounds of the DNA strand. Okay, and the first level, the simplest level of packing of the DNA is the double helix. You can see this is double helix. So in between this double helix, there are the nitrogenous bases that are actually joining and arranging, and you can say they are actually maintaining that double helix. They are maintaining the double helix. They who the nitrogenous bases, and we already know C and G do have the triple hydrogen bond between them. So there are 4 types of nitrogenous bases. You can say there are 4 types of nucleotides. C and G are making the triple hydrogen bond, and the nucleotide A and the nucleotide A and T, they are attracted and joined by the double hydrogen bond. I'm making the dotted line. Dotted line means hydrogen bond, and solid line means the covalent bond. So we are showing here by the 3 dotted line between the C and G nucleotides that are joining, that are complementary to each other, and A and T are the complementary to each other. This way, this is the, you can see the first level of arrangement. This is the second. You can see this one, another complex folding. This is another complex folding, and at the last, all these kind of foldings come across to make the chromosome. So there are different types of genes that are present. You can see this is the double helix, DNA double helix. So this is the DNA strand. So this DNA strand may have the genes, and there are different types of genes. There are different types of, you can say, genes, and there can be the different types of DNA element present in the eukaryotes. So one of the type of DNA element is a single copy gene. Some of the genes are present in the form of single copy. Some gene families are present, like 1 gene, 2 gene, 3 gene, 4, 5, 6, 7, 8, 9, 10 genes are there. They are making the similar function. They are doing the similar function. That's why they are called the gene family. They are doing the similar function. Similar function. And they are making the protein families. Gene families are making the protein families. There are the tandemly repeated genes. Some genes are repeated 2 or 3 times. 2 or 3 times tandemly repeated genes are there. 1 gene is repeated. The same gene is repeated more than one time. We will see example in the next slide. There is another DNA element type, for example, the exon and the intron. Exon and the intron. मैं पूछ लूं आवाज आ रही है?
Yes, sir.
Yes, sir.
तो आइंदा आपने ना माइक जरूर जो है ना एक बच्चे ने ऑन करना है ताकि मुझे तो पता चले कि क्या चल रहा है. मुझसे एक सुन बात करता है.
माइक पहले भी
ओके, आवाज आ रही है. ओके, ग्रेट. So another type of DNA element is the exon and the intron. So exon are those regions that actually code to make the functional product, and introns are present between the exons. For example, this box is showing the exon, and in between this exon, there is a region that is not coded to make the functional product, that is called the intron. I show by I. And there is another exon. After the transcription, the intronic parts are spliced out and the exonic parts are joined together, that are making then finally the functional product. So the exon and the intron and the intergenic DNA in between the two genes, if G1 is somewhere here in the genome, G1, and there is a long sequence, and there is then another gene G2 is present in the genome. So in between the two genes, whatever is present, the DNA sequence is called the intergenic DNA. Intergenic DNA. And mobile elements that are moving from one locus of the genome to another locus. There are some mobile DNA elements. There are simple sequences of DNA that are the simple sequences like 80, 80, and 80. 80 is repeated several times. They are called the simple sequence DNA. There can be the AGC, 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 AGC. There can be the TA, 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 TA. There can be the ATC, AGC, AGC, AGC. There are simple sequences that are repeated several times that are called the simple sequence DNA. So this is again some information here. So move to the next slide. So we are actually interested in finding the structure of the eukaryotic gene. We are interested. Eukaryotic gene. So for example, you on the right side, you can see one of the several arrangements of the genes. For example, this is the promoter region. This is the cis regulatory elements that is found near to the gene. Definitely gene is containing the introns and the exons. The blue things are the exons and the black things are the introns. Blue things will code to make the functional product, but the black part, the intronic part, will be spliced away and will not be making any functional product, will not contribute actually. So upstream of the gene, there is the promoter sequence. There is a promoter sequence. This is found nearby. For example, B is found nearby to the gene, is called the cis regulatory elements. And so there is some other regulatory element that is found almost, you can say, sometime 50 kilobase away from this promoter region. This is called the trans regulatory element sometime. So these cis and trans regulatory elements are actually contributing along with the help of transcription factors, and they make the transcription of this gene that is containing the 3 exons. These 3 exons have been translated. This is one of the structure of the gene in which the cis and the trans regulatory elements, the promoter regions, are making the mRNA from a gene. There is another type when two cis and trans regulatory elements are, for example, containing downstream of them the exon 1, 2, 3, and 4 exon containing gene. These regulatory elements are containing the 4 exon containing gene. So they are transcribing in a way that they are making the mRNA 1 that is containing all the 4 exons, 1, 2, 3, and 4. 4 exon, first mRNA is containing, but the mRNA 2 is containing the only 1, 2, and 4 exon, but the third exon have been divided, is absent. Third exon is absent. So one gene can make more than one mRNA. So this is another structure of the gene in which one gene is going to make more than one mRNA, and both mRNA are having the different exon and the different mRNA sequence, and they will make the different functional product, and they will make the different functional product. There is another scheme. There is another scheme where the cis transregulatory elements are making. There is, this is similar actually. They are making the product. For the mRNA, one is containing exon 1 and exon 2, while the mRNA 2 is containing exon 1 and exon 3. So none of the functional product is containing full length of all the exons, but both mRNAs are different and are not containing all the exons. But here, in this case, one of the exon, one of the mRNA was containing all the exons. There is another scheme. There is another scheme. Sometimes the regulation, sometimes the regulatory elements are found in between the exons. Sometimes, for example, G, the below region, that is the regulatory element, is found in between the exons, and we can see the functional product mRNA1 is containing exon 1 and 2 and 3, but the mRNA2 is containing the exon 1b, exon 2 and 3. This way, different types of genes, types of gene are making the different types of functional product, the different types of mRNA actually. Okay, we move next. All these information are have been okay, but tables are missing. Several tables are missing here. Okay, we come here. We come here. So every individual may have some of the differences in their genome. Those differences can be mapped, can be fingerprinted, just like the fingerprints of our hands. The DNA fingerprinting can find the differences between the individuals, and they can compare and they can match the different individuals that are related to each other. And this kind of specific DNA sequences in one type of individual can be used to identify and recognize that individual. So, for example, there are two cases. This is one of the paternity determination case in which mother is there. Definitely, when we want to find the who is the father of the child, so mother is always confirmed. We never are confused with the mother. Who is the mother? For example, the mother DNA is taken and then it is restricted and run on the agarose gel, for example, or the SDS page gel for more clear differentiation between the bands. This is the DNA fragments of the mother. You can see the fragmentation pattern. And this is the child's DNA fragmentation pattern. The baby that was born, his or her DNA was taken and run along with the mother. Definitely some of the DNA patterns from the mother will be appearing in the baby, in the child. And you can see this one is matching exactly from the mother and the child. And this one is again matching, and this one is again matching. And you can say this one is matching, okay? And this one is matching from the mother to the baby. And there are two of the, one of the father, for example, is claiming, or second one is also claiming, or both are not getting agree. And to find the father, we also take their DNA and run along with the mother's and the baby's DNA. And this is making the true comparison. Okay, we can see here, father 2 is containing one of the band, that is the distinguishing band that is present in the child. You can see this one. But for father 2 is containing this band specific. There is another father 2 is containing one extra band, and that is not present in the baby. Here again, father 2 is not containing a band that father 1 is containing. Father 1 means suspected. We want to know which one is father. So father 1 is containing this band that is similar between the child and the father. There is another distinguishing feature that is not even present in the mother. This distinguishing band is only present in father 1. Then again, this distinguishing band is only present in father 1, not in father 2. And again, we can see here the distinguishing band, and we can see here the distinguishing band. One, 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 two, 3, 4, 5, 6. 6 distinguishing bands are only present in father 1 and the child, but not in father 2. So this way we can by the DNA fingerprinting know that F1 is actually the father of the baby, father of the children. So this way, the paternity can be found by the use of DNA fingerprinting. And in the criminal identification, there is another case, like for example, this is the victim. We take the DNA of the victim. We take the DNA of victim, and victim to prove that which one is suspect. We take also the DNA of suspect 1, 2, and 3. We take the DNA of all 3 suspects, and we also obtained one sample from the site, uh, from the site of incidence, or you can say where the crime actually occurred. We took one of the sample and also extracted the DNA, victim's DNA, and the specimen found from the site of murder or site of rape. We obtained one sample here, and 3 of the suspect sample was also obtained, the DNA sample, and all 3 samples were processed and then run on the SGS page gel or the agarose gel. And we want to find the distinguishing band that is present between the specimen and the suspect, and that should not be present actually in the victim. That will be more, you can see, the deciding band, which it will prove that the suspect was actually present at the site of crime. The suspect was present at the site of crime, and the suspect is actually the criminal. So this kind of DNA fingerprint will tell us. Okay, let's see. For example, you can see this one is the one of the band that is present in both specimen and, you can see, the suspect. And again, we can see some band. This one is here. This one is matching here. This one is again matching here. This one is again matching here. And we can see the suspect 1 bands are more matching with this specimen. It means the specimen found from the site of crime is the specimen of the suspect 1 actually. So we can, from this DNA fingerprinting data, know that the suspect 1 was actually present at the site of crime, and maybe the suspect 1 has committed the crime. Suspect 1 has committed the crime. This way, the criminal identification is facilitated by the DNA fingerprinting. Okay. Okay, so this structure has already been seen by us. So you can see the nucleosomes are here, and they are found in the zigzag format. They are saying zigzag ribbon. The nucleosomes are making, and these zigzag ribbons are then making this kind of solenoid model of the nucleosomes. And these nucleosomes are shown in another format, and this is the, you can say, the surface contour image. This is the cartoon picture actually, and this is the surface contour image that is showing the different nucleosomes and their packing actually. So here we already know there are 4 types of histone proteins: H3, H4, H2A, and H2B. There are 4 types of nitrogenous bases, and they are having that, you can see, some of the C-terminal tails are coming out, and the histone proteins are actually acetylated, and their acetylation actually facilitate the transcription of the genes. If the acetylation of the RAS gene happens, RAS gene, for example, RAS is the gene that is overexpressed, and then it causes the lung cancer. If it is activated more and more, it means there will be lung cancer. So in the case, for example, the the promoter region and the gene that is containing the histone proteins, if they are acetylated, the more RAS protein will be made, more transcription will be there, more RAS proteins will be made, and there will be the lung cancer. For example, if there is the histone acetylation of the tumor suppressor gene, so RAS is the cancer causing gene, and I write the name of a tumor suppressor protein, the p53. This is very, very famous, p53, very famous protein. Maybe in whole of the world, the most of the work was performed, mostly much work is performed on this single gene, much more work as compared to any other gene. Maybe I guess. Okay. So
Sir,
if
this in
the
paper.
acetylation, histone acetylation for this gene occurs, then there will be more transcription, and there will be more p53 protein, and the tumors will be suppressed. So histone acetylation actually increases the transcription, and the more protein is thus made. If we talk about the DNA methylation, okay, If we talk about the DNA methylation actually decreases the transcription. In some of the cases, the tumor the tumor suppressor gene have the higher methylation. Thus, tumor suppressor genes are less expressed, and less tumor suppressor protein is made. Thus, there is a cancer. Sometimes the tumor suppressor gene are suppressed by the over methylation of the DNA. Thus, methylation inhibits the transcription. So some of the figures are still there. I actually open from the PDF. I opened from the PDF chapter and I show you the more picture. नेक्स्ट टाइम एक बच्चा माइक जरूर लगाए. मैं एक दफा अटेंडेंस ले लूं आपकी. अटेंडेंस देख लूं कितने बच्चे आए हैं. अभी तो 40 स्टूडेंट आ
Yes,
गए
sir.
जी
माशाल्लाह.
सर.
बहुत अच्छी बात है. अटेंडेंस यहाँ से ले लेते हैं.
सर आवाज
सर
नहीं आ
हम
रही.
2 स्टूडेंट क्लास.
सर आपको हमारी आवाज नहीं आ रही.
सर हम 2 स्टूडेंट से इकट्ठे क्लास ले रहे हैं.
ये PDF चैप्टर नजर आ रहा है बच्चों को आपको?
यस सर. ये
ओके. तो मैंने GR से पूछ लिया, उनका शुक्रिया. शायद कोई टेक्निकल प्रॉब्लम, मुझे आवाज आपकी नहीं आ रही, लेकिन आपके माइक ऑन है. तो PDF चैप्टर आपको नजर आ रहा है. दिस इज गुड न्यूज. हमने ये फिगर पढ़ ली है पहले, ठीक है. और इसके बाद ये ना डिफरेंट किस्म के ना एक्सॉन डुप्लीकेशन कैसे होती है. Some of the time exons are duplicated. So this is the L1 region, that is actually the homologous region. Homologous mean containing the similar sequence or same sequence. For example, these are the parental chromosome, one coming from the father, one coming from the mother. If this homologous region is very much similar from the mother to the father, there can happen an event in which the homologous recombination actually duplicate the exon 3 on one of the chromosome. So when the gamete will be made, one of the gamete will be containing the duplicated exon 3, and the another gamete will be containing the gene without the exon 3. Thus, there is a chromosome, there is an arrangement of the exons that are duplicating actually the exons. And this process by the homologous recombination is called the recombination. And this is called the unequal crossing over, because one is getting the exon, another is losing the exon. This is called the unequal crossing over. And the gene duplication, how the genes are duplicated, these are the parental chromosome. For example, the beta globin gene is here. For example, beta globin gene, maybe it is containing the 3 exons shown by the green blocks. And by the homologous recombination, one of the chromosome is getting the beta globin gene, another is losing the beta globin gene. This way, the homologous, again, the recombination is duplicating the gene, and one of the gamete will be containing the two copies of beta globin gene, and another gamete will be without the beta globin gene, and thus one of the baby may be getting the two of the genes of the beta globin. So another event, we have seen these kind of simple transcription unit, complex transcription unit we have seen, and we have seen also mRNA 1 and 2 are different. And we have also seen where there is the regulatory elements somewhere falling in between the exons, like exon 1A and exon 1B is containing in between them the G element, the blue block, the regulatory element. And thus, two of the regulatory elements that are falling in between the exons are making two types of functional product, two types of mRNA actually. So we have seen already this table. So protein coding genes may be long. How long? 0.5 to 2200 kb long. There are 21,000 genes in the human, and their genes are making 40% of the human genome, and rest of the genome is not making any functional product. But every gene is containing the intronic region, thus if we count only the exonic region, only 2% will be actually the functional region, and rest of the region of the human genome is the junk region, or they are not making any functional product. But it is believed that the junk region and the region of the DNA, human DNA, that is not making any functional product is actually maybe involving in the regulation of the rest of the gene, of the rest of the DNA. Long non-coding RNA, they may be containing 0.5 to 50 kb long. There are 10,000 of these kind of genes, and they are making 0.9% if we ignore the intronic region. U2S are a small nuclear RNA that supplies actually the intronic region. These kind of, so they are 6.1 kb long. Usually there are 20 genes of this kind, and they are making less than 0.0001% of the human genome. And there is a ribosomal RNA, 43 kb long they can be, and there are 300 of these kind of genes, and they are making 0.4%. And sometimes repetition is there in the DNA. Simple sequence repeats are that much long, and interspace repeated mobile elements, actually not interspace, interspersed, there are mobile elements. There are DNA transposons are 2.3 kb long, and LTR retrotransposons are 6 to 11 kb long. And that is enough. Actually, you do not need to memorize any value. Maybe maximum upper top two values may be memorized by you. And eukaryotic human, these two are the eukaryotic chromosomes, for example, are the DNA elements, and the human genome is containing much more the junk region, the non-coding region, as compared to the chromosome 3 of the of S cerevisiae. They are containing the less junk region. The greens are the exonic region here, both in the yeast and in the human. And but the human is containing a lot junk region, but the yeast is containing less junk region. Junk region means non-coding regions. Again, the non-protein coding RNAs and their function. These are some of the RNAs that are not coding for the protein, that are not meant to code for the protein. Like the ribosomal RNA are almost 300, and transfer RNA, small nuclear RNA, U7 small nuclear RNA, small miRNA, RNAs. So there are different names. You can just read, and you don't need to memorize. And this is the DNA fingerprinting application in the paternity determination and in the criminal identification. And this is the example of transposons. For example, there are DNA transposons and there are retrotransposons. DNA transposons are simply in a way that the DNA transposons are just like the copy and paste from one place in the genome. The transposon element is cut and is pasted in at another place in the genome. The gray region is showing one site in the genome and the black region is showing another site. So the gray region is cut with the DNA transposon and the black region is pasted with the DNA transposon. But the retrotransposons are different from the DNA transposons. This is the copy paste mechanism. Copy paste mechanism. From the gray region, the mRNA is transcribed and the RNA is converted into the DNA, and then the DNA part is then transferred to another place in the genome. So original place is containing the same transposon, but the new region also, new site also is containing. So this is the copy and paste mechanism. Who? The retrotransposon. Retrotransposon is involving the RNA intermediate, but the DNA transposon is not involving the RNA intermediate. So rest of the thing, one or two slides you have already seen. तो मेरे ख्याल इतना काफी है. तो इसी में से क्वेश्चन आ जाएंगे आपको इंशाल्लाह. ठीक है. डीएनए एस्टाइलेशन आ जाएगा, हिस्टोन एस्टाइलेशन आ जाएगा, डीएनए मेथिलेशन आ जाएगा. ठीक है. तो यही आपको आएंगे. ठीक है. आपके आने का शुक्रिया. मैं देख लेता हूँ कितने लोग हैं आप. 43 हैं. मैं एक बार फिर अटेंडेंस ले लेता हूँ आपकी. तो आपका शुक्रिया. आप जो है ना पेपर में आइएगा. पेपर उम्मीद है आसान होगा. मार्किंग भी अच्छी होगी आपकी और कोई प्रॉब्लम नहीं होगा आपको. डाउनलोड अटेंडेंस लिस्ट. ठीक है. ठीक है. शुक्रिया.
ओके सर.
ठीक है जी. बाय बाय. अल्लाह हाफिज. मैं मीटिंग एंड कर रहा हूँ. एंड मीटिंग. थैंक यू.
ओके, अल्लाह हाफिज.
अगर सामने